Hey guys, welcome to AP Biology, and we're going to be talking about membrane transport part 2 today, and it's all about getting to the other side no matter what. Okay, so let's go ahead and start. All right, so we're going to talk specifically about how substances do cross the cell membrane. Okay, and it depends upon several different factors. Okay, remember the cell membrane is a selectively permeable membrane, otherwise known as SPM. So, whenever you see SPM, we're going to be talking about a selectively permeable membrane. And specifically, at this point, we're going to be talking about small molecule transport across the cell membrane. So the permeability of the membrane depends on depends on the molecules and depends upon certain properties of those molecules. Number one, size. Small molecules pass easier and faster than do large molecules. Polarity. Small nonpolar molecules travel faster. Okay. Charge. Ions, anything with a charge, positive or negative charge, will not cross that cell membrane. And finally, the concentration gradient. The greater that difference is between the inside and the outside, the concentration, like say there's a really super high concentration of sodium on one side of the membrane, which is an ion, and a low concentration of sodium on the other side of the membrane, well, then sodium can travel through the membrane through a special protein. It cannot diffuse simply through the membrane, but the concentration gradient will make it move faster. Okay, so let's look at this particular diagram on the right here. So hydrophobic molecules. These are molecules that do not have a charge, i.e. they are nonpolar. Oxygen, CO2, nitrogen easily pass through the membrane, and we can see that there is a large, thick arrow showing that. Small, uncharged polar molecules. Okay, so here we have water and glycerol. Now, it shows that those can pass through the membrane, but I'm going to tell you right now that water also has a special protein that just transports water. It's called an aquaporin. Okay, pore means like a pore in your skin. The IN after it indicates it's a protein. So it's a water transport protein, aquaporin. And that makes water move a lot faster through the membrane than simple diffusion. Okay, large uncharged polar molecules such as glucose and sucrose will not cross the cell membrane. Ions, which have a charge like chlorines, potassium, and sodium, anything with a charge, will not cross the cell membrane, at least by simple diffusion, which just means moving through the phospholipids. They will not do that. They can get across, but they have to use specialized transport proteins. Okay, so we can divide membrane transport into two major categories: passive transport, which requires no energy, and active transport, which requires energy in the form of ATP. So think about passive transport. It's a movement from areas of high solute concentration. Those little square brackets around high solute mean concentration of. So the way I read that is high solute concentration, to areas of low solute concentration, i.e. passive transport only works going down the concentration gradient from high to low. That's like a rock rolling down a hill. Active transport, which requires energy in the form of ATP, is the opposite. It's the movement from areas of low solute concentration to areas of high solute concentration. You're going against the concentration gradient. That's like a salmon swimming upriver or you pushing a rock up a hill. You have to put a lot of energy into it in order for that to happen. Okay, so diffusion is simply the movement of molecules from an area of high concentration to an area of lower concentration. That can happen in the air. Just think of somebody wearing perfume. Somebody puts perfume or cologne on, you can smell them, and it gets really intense when you get close to them. I know there's a couple of people in this school that must take baths in this stuff or something, because I can smell them from a mile away, but as I get closer, that intensity gets even stronger. Okay, and here the example is just showing you the random movement of taking like a sugar cube, which most of you guys are probably not familiar with, dropping it into a glass of water, and those sugar molecules will eventually disperse evenly throughout that glass of water. Or you take a drop of food coloring, drop it into a glass, and just watch what happens. It starts to move all around until eventually that whole glass is colored blue. It's that happens anywhere. It does not have to be a living system. It can be a completely abiotic system, and diffusion, simple diffusion, will happen. It can be in water, it can be in the air, so on and so on. And it will continue until equilibrium is reached. What does that mean? It means that there's no net movement in any one direction. So here the net movement is going to be away from the area of concentration. Here the molecules are going to be moving randomly all over the place, but not into one single direction. Okay? Again, so passive transport is diffusion across a membrane. In passive transport, the substances diffuse through the membranes without any energy input by the cell. So here we got like molecules of dye. We have a semi-permeable membrane here in the middle, and you can see that these dye molecules are moving randomly, but eventually they will go through this, little holes in here, it'd be like dialysis tubing like we used in lab, until there's an equal number of those molecules on either side. Now, will they stop moving? Of course not. It's just that there's going to be no net direction of movement. Okay, so all the molecules are not going to go to one side versus the other. They're going to be all evenly dispersed. And now the important thing to remember about passive transport is that each solute diffuses down its own concentration gradient. So what we see here in the first picture in the bottom left here is that we have green on the left and purple on the right. So higher concentration of purple on the right, higher concentration of green on the left. These molecules are going to move randomly until they are both equally distributed across the cell membrane. So remember that, because I see a lot of questions that will talk about aspects of that. Simple diffusion, just moving down the concentration gradient. Facilitated diffusion is also similar to simple diffusion, except that it's a type of diffusion that occurs with molecules that cannot simply diffuse across the cell membrane, i.e. they have a charge like an ion, they're polar, they're large. Those are all types of molecules that cannot simply diffuse across the cell membrane. They must use a transport protein. Okay, but similar to simple diffusion, it's also facilitated diffusion does not require energy. It is a form of passive transport. It's always going to go down the concentration gradient from an area of high concentration to an area of low concentration. And there are three essential characteristics. It's very specific, meaning that a glucose transport protein will only transport glucose. A sodium ion transport protein will only transport sodium ions and not chlorine, and so on and so on and so on. It is passive, meaning it does not require energy, and it will go as fast as there are in terms of the number of proteins. It'll reach a point at which every single transport protein is moving the, let's say, sodium ions across the membrane as fast as it can. That is the highest it will occur. It can't go any faster than the number of transport proteins you have. So facilitated diffusion will continue increasing its rate until every single protein has become saturated, and therefore a rate increase will not happen after that point. Okay? So again, like I say here, each polar molecule is transported across the plasma membrane has its own type of channel. Channel's a protein, and as I said earlier, the permeability is highly selective. Sodium-only transporters only transport sodium, so on and so on. Okay? Ions move across the membrane in ion channels, which are also proteins. So again, you have a chlorine transporter, a sodium transporter, a potassium transporter, so on and so on. And these will become very important when we talk a little bit about how your nervous system works, especially the sodium and the potassium channels. So here's just a picture of a carrier protein. This is the high concentration of the orange blocks outside the cell. Inside the cell, it's a low concentration. And this is going to be the movement of this molecule through a transport protein. So this is a type of facilitated diffusion. And the carrier proteins basically will bind to the molecule and then bring it across. And there's actually several different methods by which that can happen, and it is driven by concentration gradients. Now, osmosis, kind of what we did in lab, all right? We used dialysis tubing. Inside the dialysis tubing, we had a solution of sucrose. Sucrose is a disaccharide. The dialysis tubing has small little tiny holes in it. The disaccharide, i.e. sucrose or table sugar, is too large to fit through those holes. But water can fit through those holes. And here's the phrase you want to meet. Remember, water chases the solute. Okay, so wherever the solute concentration is greater, water will move to it. So in an aqueous solution, i.e. a solution of water, both water and solutes diffuse down their own concentration gradient. Okay, most of the solutes that we're going to be talking about are not lipid soluble. That means they cannot simply diffuse across the membrane. Okay, they have to go through a transport protein. Water is one of those. Now, water can leak across that membrane, but it has its own special transport protein called an aquaporin, as I mentioned earlier. Okay, and that movement of water across a membrane is called osmosis. So whenever we're talking about osmosis, we're talking about the movement of water, not solute particles, water. Okay, so we need to have some more terms here. Okay, osmotic concentration. That is the concentration of all the solutes in a solution. Hyper means above. So a hypertonic solution is a solution that has a higher solute concentration. Hypo means below or under. A hypotonic solution is one that has a lower solute concentration compared to another one or compared to another side of the membrane. Isotonic, they're both equal. Okay? And when we talk about how water moves, this is also important to recognize. Water moves always from a hypotonic environment to a hypertonic environment. And in that, we are specifically referring to the solute concentration. So where there's a low solute concentration, there's a high water concentration. Where there is a high solute concentration, there is a low water concentration. So even though water is moving from hypotonic to hypertonic in relation to the solute, water is moving down its own concentration gradient from a high water concentration to a low water concentration. Okay, so that might be a little confusing, but we'll hopefully clear that up in class if you have questions on it. Okay. Oops, skipped around there. So how does that apply to cells? Well, let's start off with an animal cell. If I take an animal cell and I put it into an isotonic environment, okay, that where there's some salts on inside and outside, water will move in and out of that cell, and that cell will be in its normal environment, maintain its normal shape, do its normal functions. If, however, I put an animal cell, such as your red blood cells, into a hypotonic solution, i.e. a glass of distilled water, water is going to chase the solute. There's more solutes inside of the cell, so water will move from the environment into the cell, and because animal cells do not have a cell wall, that will cause that cell to explode, lyse. So if you are ever super dehydrated and you have to go to the hospital and they have to put IV fluids into you, they would never, ever put pure water in because that would kill you. What they put in is a saline solution. It's like 10% salt, 8 to 10% salt, okay, because that is what is isotonic with your blood, all right? Now, if you put a red blood cell, animal cell, into a hypertonic solution, one that has lots of solute, so there's more solute in the environment than there is inside the cell relatively, water will move out of the cell into the hypertonic solution. Okay, and that will cause the cell to shrink and shrivel. Plants are a little bit different. Plants have a cell wall. Okay, so plants like to be in a hypotonic solution. That means water will rush into the plant cell. Okay, and that's what plants want. They have that big, large central vacuole that will fill up with water, and it will push against the cell walls and make that plant cell turgid, rigid. Okay? When you forget to water your plants, what starts to happen? It's the water starts moving, starts becoming isotonic, so water starts moving in and out of that cell. There's no longer any turgor pressure. That's what that pressure inside the cell is called, turgor pressure. That gets less, and the cell walls begin to collapse, and the plant begins to wilt. Okay? Put a plant into salty soils or into salt solution, and then those cells are going to basically lose all their water, and the cell membrane will actually peel away from the cell wall. That's called plasmolysis, and that destroys the cells. I'm hoping we can observe that under microscopes next week. So we'll look at that hopefully next week. So plants like to be in generally a hypotonic environment. So let's look at this picture here. I have a special glass apparatus that has a selectively permeable membrane on one side and a 3% salt solution. I put it into distilled water. The distilled water is going to be hypotonic in relation to the salt solution, which is hypertonic. Remember, water chases the solute. Water goes from hypo to hyper. So water is going to move in through this selectively permeable membrane because the salt solution cannot get out of it. And it's going to cause that, it's going to cause the salt solution to rise and become less dilute. Okay, or become dilute, not less dilute. All right, so that's what's going to happen there. And that is it for this particular slideshow. And we'll get into water movement on our next class.